Hi, hello to all the NEET aspirants. Hope you all are doing great and preparing well for your exam. Now we are going to discuss the most recent paper, that is NEET 2024 botany paper, in a more interesting and a unique way. Okay. So if you look into the first question, this is from biological classification, wherein rhizopus which belongs to Phycomycetes is a bread mound. Okay, rhizopus is a bread mound, is commonly called as bread mound. Next is Eustylago. Eustylago is a smut fungus. Puccinia is rust fungus. Agaricus is mushroom. Agaricus is a type of mushroom which belongs to Basidiomycetes. So if you look into the correct answer, A is 3, B is 2, C is 4 and D is 1. Option 1 is the correct answer. Okay. Next question, if you see beta, next question, see all the diagram based questions also we are going to cover. Stay tuned. In this question, the question is from monocot seed of morphology of flowering plants, wherein in a monocot seed, I have explained you multiple times, but then again I am saying this is the endosperm. Okay, endosperm, wherein here you have a papery cotyledon called as scutellum, and in this embryonal axis the above part is plumule. Covering the plumule there is a protective sheet, that is coleoptile, and here at the C region radical, covering the radical the protective sheet is coleorhiza. So here he is asking us which is destined to form root. Radical is destined to form root, we all know that. Here C exactly locates radical and D locates coleorhiza. Don't get confused at all. Look into the proper locations of each and every region in the diagram, then you can easily tackle the question beta. So here what is the correct answer? Option C is the correct answer, that is option 3. Okay. Next, if you see lecithin, third question, that is lecithin, a small molecular weight organic compound found in the living tissues, is an example of, this is an example of phospholipids. Okay, this is an example of phospholipids whose lipid composition is high in the cell membrane. Okay, which lipid composition will be high in the cell membrane beta? That is phospholipid. Okay, do remember. Next question, this is from molecular basis of inheritance or MolBio, wherein a transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream. Okay, upstream and downstream, how do you locate it beta? Upstream is from 5 prime of the coding strand or the mRNA to 3 prime of the coding strand or mRNA. This 5 prime and 3 prime is located with respect to coding and mRNA only. Okay, coding strand and mRNA. Okay, so these two, coding strand and mRNA, at 5 prime region promoter is located, at 3 prime region terminator is located. In between promoter and terminator we have structural genes. Okay, we have structural gene in between promoter and terminator. Clear, Nana? So now if you see, the transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream, that is promoter, structural gene and terminator, which is option 4. Clear? Yes. Next question, if you look into, again match the following type of question from genetics, that is principles of inheritance. So here if you see, Nana, 2 or more alternative forms of gene, are called as alleles. Only 2 alternative forms will be in a usual, but those in multiple allelism you can see more alternative forms of a particular gene. Best example is what, Nana? ABO blood grouping for multiple allelism. Okay. Next, cross of F1 progeny with homozygous recessive parent. Okay, with homozygous recessive parent is called as test cross. Okay, this is what? Test cross. Cross of F1 generation with any parent, that is called as back cross. Next, number of chromosome sets in a plant. complete set of chromosome in an organism is diploid. We'll call that as diploid. Half the set of chromosomes is haploid. So, number of chromosomes is deciding the ploidy of an organism, right beta? So, now what is the correct option if you see? A is 2 or more alternative forms, that is A is 3, B is 4, C is 1, D is 2. Option 3 is the correct answer. Next question if you see, auxin is used by gardeners to prepare weed free lawns, but no damage is caused to grass as auxin. Okay, first, to promote weed free lawns, what type of auxin we are using? We are using synthetic auxin. We are using synthetic auxin, that is 2,4-D. That is 2,4-dichlorophenoxyacetic acid. Okay, 2,4-dichlorophenoxyacetic acid we are using to promote weed free lawns. Okay, so this auxin is not damaging monocotyledonous plants. Okay, as this doesn't affect monocotyledonous plants, we are using that, and this causes major effect to dicotyledonous weeds. This causes major effect to dicotyledonous weeds. So, what is our answer here with option 3? Okay. Next, the capacity to generate a whole plant from any cell of the plant is called as totipotency. The capacity of generate the whole plant from one cell is called as what, Nana? Totipotency. Next, this is from microbes in human welfare, wherein Clostridium butylicum, this will produce butyric acid. Saccharomyces cerevisiae will conduct anaerobic respiration. What respiration, Nana? Anaerobic respiration. It will conduct anaerobic respiration, and this will produce ethanol and carbon dioxide. This will produce ethanol and carbon dioxide by conducting alcoholic fermentation. Okay, so the end products of alcoholic fermentation by yeast are ethanol and carbon dioxide. Trichoderma polysporum will produce cyclosporin A. Streptococcus will release streptokinase. Okay, so here the right answer will be A is 3, B is 1, C is 4 and D is 2. So, option 3 is the correct answer. Next question, if you look into spindle fibers attached to kinetochores of the chromosomes during spindle fiber attachment to the kinetochores. Look into it properly, beta. Kinetochores are the disc shaped structures which are around the centromere. Okay, so spindle fibers are connected to the kinetochores of the chromosome at metaphase stage. At what stage? Metaphase stage of cell division. In this stage only spindle fibers are connected to the kinetochores of the chromosome that aligns all the chromosome as a equatorial plate, and that will further move into the centromere splitting in the anaphase. Okay, so the attachment of kinetochores to chromosomes will happen in which phase, Nana? It will happen in the metaphase. Option 2 is the correct answer. Which of the following is an example for actinomorphic flower? Best example for actinomorphic flower is Datura. Okay, rest, Pisum, Cassia, Sesbania, they will have zygomorphic flower. All the time in all the previous year questions, you got either 1 or 2 questions for sure from morphology examples itself. So, focus more, once again revise more with respect to morphology examples once again. Okay. The cofactor of the enzyme carboxypeptidase is zinc. Okay. The cofactor of the enzyme carboxypeptidase is what, Nana? Zinc. That is from biomolecules, right? Next, this is from Cell the Unit of Life, nucleolus. Okay. Nucleolus is the site for rRNA synthesis. That is A is 3. Next, centriole. Centriole has an organization of cartwheel with what arrangement? 9+0 arrangement. Leucoplast will store nutrients. Okay, Golgi bodies are the best site for the synthesis of glycolipids and glycoproteins. So here if you clearly look into beta, A is 3, B is 2, C is 4 and D is 1. Option 1 is the correct answer. Next question, if you look into, identify the set of correct statements. The flowers of Vallisneria are colorful and they produce nectar. Wrong, because Vallisneria and Hydrilla, they are conducting what type of pollination beta? Hydrophily. Okay, they are conducting hydrophily. So those plants whose pollination method is either through wind or water, they don't have colorful petals or neither they produce the nectar. Okay, so that is wrong. Next, the flowers of water lily are not pollinated by water. True, water lily, water hyacinth, they are not pollinated by water. Next, the most water pollinated species are the pollen grains are protected from wetting. True. Next, pollen grains of some hydrophytes are long and ribbon like. Exactly true. Next, in some hydrophytes the pollen grains are carried passively inside water. Correct. So if you see what are the correct statements ma? B, C, D, E. Option 4 is the correct answer. Next, if you see Nana, statement based question that is from plant kingdom. Parenchyma is living but collenchyma is dead. See, this question is connecting two chapters, that is anatomy and plant kingdom. Watch carefully. Parenchyma is living and collenchyma is dead. Wrong. In 3 simple permanent tissues, that is, what are the three simple permanent tissues? Amongst 3 simple permanent tissues, those are parenchyma, collenchyma and sclerenchyma. Okay, parenchyma, collenchyma and sclerenchyma. Amongst these 3, parenchyma and collenchyma are living and sclerenchyma is dead. Okay, parenchyma and collenchyma are living and sclerenchyma is dead. So here now you answered this question, parenchyma is living, collenchyma is also living. So this statement is wrong. Next, gymnosperms lack xylem vessels. True, but the presence of xylem vessel is characteristic feature of angiosperm. Gymnosperms lack xylem vessels. They also lack sieve tubes and companion cells from phloem. They don't have sieve tubes and companion cells. Instead, they have sieve cells and companion cells. They have sieve cells and companion cells. Sorry, albuminous cells. They have sieve cells and albuminous cells. Clear beta? Understood? So now here statement 1 is wrong and 2 is true. Option 4 is the correct answer. Next, the lactose present in the growth medium of bacteria is transported to the cell by the action of permease. Quickly recall the Lacoperon model. In Lacoperon model, what do we have, Nana? From 5 prime to 3 prime. From 5 prime to 3 prime, we have promoter, then followed by I gene, followed by promoter and structural genes Z, Y, A, lastly it is terminator, right? So to promoter region, always RNA polymerase is coming and binding, right? To promoter region, always RNA polymerase is coming and binding. Next, I gene. I gene will release or secrete out repressor. Okay, I gene will give out repressor. Next to the promoter region again, RNA polymerase will bind. Okay, RNA polymerase will bind. Next, Z gene. Okay, Z gene will give out beta galactosidase, is coding for beta galactosidase, Y for permease and A for transacetylase. Beta galactosidase will cleave the disaccharide into 2 monosaccharides, glucose and galactose. Permease will increase the permeability of lactose into the cell. okay, and A is trans-acetylase that will make the monosaccharides more soluble in the cell by changing the group, okay. Now, if you see which in the growth medium of bacteria, lactose has to be transported to the cell more by the action of what, Nana? Permease, which is coded by Y gene, okay, do remember that. Next, formation of intervesicular cambium from fully developed parenchyma, okay, is the best example for what? De-differentiation. That is the best example for de-differentiation. How? Let us quickly remember all the tissues. First, primary meristematic tissues include apical and intercalary meristems, right? Apical and intercalary meristems. Apical meristems include shoot apical meristem and root apical meristem, right, Nana? Next, this will form primary plant body. The meristematic activity of this primary meristematic tissues will form primary plant body with permanent tissues, okay. Those permanent tissues are differentiated, right? They are differentiated and they are of two types: simple permanent tissue, complex permanent tissues. Simple permanent tissues include, just now we have discussed, parenchyma, collenchyma and sclerenchyma. Complex permanent tissues include xylem and phloem. Clear, Nana? Now, amongst this, after the formation of primary plant body, plant to increase its girth or size, okay, it will conduct secondary growth. For that, in cortical region, few of the cells will get meristematic, forming out cork cambium. They will form out cork cambium, and in the stellar region, in between xylem and phloem and in between two vascular bundles, cambium is formed, which is called as vascular cambium. These are gaining back the meristematic activity again. They will perform cell division activity, right? So, these are de-differentiated tissues. They lost the capacity of division and gaining back the cell division activity. So, they are called as de-differentiated tissues. They include cork cambium and vascular cambium, okay, which is then followed by redifferentiated, as they will conduct the division activity and they form new cells. Those new cells are what, Nana? Secondary cortex, cork cells in the cortical region, secondary xylem, secondary phloem in the stellar region. So, these are again redifferentiated tissues. What are redifferentiated tissues, beta? Secondary xylem, secondary phloem, secondary cortex and cork cells, okay. So, do remember this. So, here if you see intervesicular cambium, and which is a vascular cambium in between two vascular bundles, this is formed as de-differentiated tissue, okay. Next, inhibition. So, any of the tissues whichever they are asking you in the exam, okay, either redifferentiated, de-differentiated or differentiated, don't get confused at all, okay. These are the tissues with respect to that, do remember. Next question, if you see, Nana, inhibition of succinic dehydrogenase enzyme by malonate is classified under competitive inhibition, okay, in ETS, okay, electron transport system of mitochondria from complex 2. Complex 2 is succinic dehydrogenase, right? So, that is competitively inhibited by malonate, okay. To remember, this is also asked in one of the previous year question again. Next, diagram based question again from morphology, okay. So, here if you clearly see, Nana, identify the type of flower based on the position. We know three types of flowers, right? What are they? What are the three types of flowers we know? Hypogynous, perigynous and epigynous. Hypogynous, perigynous and epigynous flowers, right? So, hypogynous flowers have superior ovary. They have superior ovary. Perigynous have half inferior and half superior ovary. Half inferior and half superior ovary. Okay. Epigynous flowers include inferior ovary. Inferior ovary. If you look into this type of flower, where ovary is half inferior and half superior, rest of the floral parts are at the rim of the thalamus. In both the diagrams. So here what will be your answer, Nana? Both are of perigynous flowers. Okay, both are of perigynous flowers. Do remember. Next, in a plant, black seed color is dominant, both homozygous and heterozygous dominant over white, which is of this. In order to find out the genotype of black seed plant, okay, whether it is homozygous or heterozygous, with which of the following genotype you will cross. Always to know the genotype of the organism, we will do test cross, right? What do we conduct, Nana? Test cross. We will do test cross. Test cross in between dominant phenotype and recessive parent. So this is the dominant phenotype, it has to be crossed with the recessive parent, that is this genotype. Okay. Then this is again from principles of inheritance. Next question. This is from plant physiology, wherein in the given figure, which component has thin outer wall and highly thickened inner walls is the question. So here if you clearly see, Nana, these are subsidiary cells over here, B is subsidiary cells. Next, these are guard cells, okay, guard cells of the stomata. These are guard cells of the stomata. They are of bean shaped, so it is a dicotyledonous leaf, and guard cells have thin outer wall and thick inner wall, thin outer wall and thick inner wall, and here at the center, that is the stomatal pore. Both stomata, guard cells and subsidiary cells, they will form stomatal apparatus. So if you see clearly, Nana, they asked us which component has thin outer walls and highly lignified inner walls, that is guard cells. Here if you clearly see what is the labeling of the guard cell, that is C. So here the correct option is option 1. Next, if you look into, this is from cell biology, chromosomes become gradually visible under light microscope during liptoting stage. True. Next, the beginning of diploting stage is recognized by dissolution of synaptonemal complex. True. We have 5 substages under meiosis 1, those are liptoting, zygotine, pachytene, diploting and diakinesis. In liptoting, chromosomes are lightly visible under the light microscope. Synaptonemal complex, which is formed in the zygotine, is dissoluted after crossing over, which is happening in pachytene. That dissolution happens of synaptonemal complex is in diploting stage. This is true. So both the statements are true. Okay, both statement 1 and statement 1 are true. If the question is about dissolution of chiasmata, okay, dissolution of chiasmata, then your answer should be diakinesis, that is the last stage of prophase 1. Clear? Yes. Next, which of the following are required for dark reaction of photosynthesis? For dark reaction of photosynthesis, of course, we need the assimilatory power, which is the last step of light reaction, that is ATP and NADPH. And of course, we need carbon dioxide for fixation, that is coming from the stomata, okay, in the gaseous exchange. So here for dark reaction to be conducted, we need carbon dioxide, ATP and NADPH, that is C, D and E. Clear? Next, which one of the following can be explained on the basis of Mendel's law of dominance? From Mendel's law of dominance, we have to pick out the correct statements. Out of one pair of factors, one is dominant and the other is recessive. True? Yes. Alleles do not show any expression and both the characters appear as such in F2 generation. Wrong. Alleles show the alternative expressions of the gene. So this is wrong. Factors occur in pairs in a normal diploid plants. True. Factors are genes according to Mendel. Next, the discrete unit controlling a particular character is a factor. Next, the expression of only one of the parental characters is found in a monohybrid cross, true. So, what are the correct statements here, Nana? A, C, D and E. Okay. So, option 2 is the correct answer. Next question, which one of the following is not a criteria for classification of fungi? Definitely it is mode of nutrition, because mode of nutrition is common for all the fungi, that is heterotrophic, right? So, classification is based on morphology of mycelium, mode of spore formation and fruiting body. Okay. Next, bulliform cells are responsible for, okay, where do you find these bulliform cells? In the monocot leaf epidermis. These are the large cells than the epidermal cells. These bulliform cells will maintain the shape of the leaf both in turgid and flaccid conditions. So, yes, it will the inward curling of leaf during flaccid conditions is shown in the monocot. So, the correct answer is option 1. Okay. Inward curling of the leaf is shown during flaccid condition only in the monocot plants, because bulliform cells are present in the epidermis of monocot leaf. Next, pink flowered snapdragon plant was crossed with red colored snapdragon plant. What type of phenotype is expressed in the progeny? Okay. So, pink color flower, this is from incomplete dominant example, you know that. So, pink color flower is crossed with red color flower. You tell me, Nana, what will be the offspring ratio? Of course, it will be 1:1, where you will get both red as well as pink colored flowers, that is option 2. Next, how many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in Kelvin cycle? Of course, during carboxylation no ATP and NADPH are required, but for reduction, for every carbon dioxide molecule, 2 ATP and 2 NADPH are required. Okay, that is during reduction step, and during regeneration it requires 1 ATP. So, on total how many ATP beta? 3 ATP and 2 NADPH. So, on total it is 3 ATP and 2 molecules of NADPH. 3 molecules of ATP and 2 molecules of NADPH are required for 1 carbon dioxide fixation. Next, identify the correct description about the given figure. This is again from sexual reproduction of flowering plants, wherein this is having long tassels. This is nothing but the diagram of long tassels of corncob, wherein the wind pollinated plant inflorescence will show the flowers with well exposed stamens. Of course, this is having well exposed stamens, and of course, this is wind pollinated plant. Okay. So, here it is option 1. So, you got diagram based questions from morphology and sexual reproduction last time. So, look into it more keenly beta. Next, Frederick Griffith has given us the transformation principle. Next, Jacob and Monod gave us Lacoperon model, that is gene expression. Hargobind Khorana gave his contribution in genetic code. Michelson and Stahl gave the experimental proof for semi-conservative mode of DNA replication. So, here if you clearly see, Nana, A is 3, B is 4, C is 1 and D is 2. Option 2 is the correct answer. Next question, spraying sugarcane crop with which of the following plant growth regulators increases the length of the stem, thus increasing the yield. Okay. So, on spraying, what you will increase the internodal length? Immediate plant hormone or phytohormone comes to our mind is gibberellin. Okay. On spraying gibberellins, internodal length will be elongated in a stem. Next, from list 1 and list 2, if you suppose, rose, Plum, rose, peach will have what type of flower? Perigynous flower. Best examples for perigynous flowers are plum, rose, peach. Next, P has marginal placentation where all the ovules are arranged on the ventral suture of the ovary. Okay. Next, cotton shows twisted aestivation. Mango has drupe type of fruit. Now, if you clearly look into the options, A is 4, B is 2, just a second beta, A is 4, B is 3, C is 2 and D is 1. No, just a second. A is 2, P that is having marginal placentation 4, C is 1, D is 2. Option 1 is the correct answer. Next question, read the following statements and choose the set of correct statements. Okay. So, and in the members of Phaeophyceae they are asking Phaeophyceae, that is brown algae, that is brown algae. Asexual reproduction occurs usually by biflagellate zoospores. True. Sexual reproduction is oogamous. Wrong. Because it can be isogamous, anisogamous and oogamous. Stored food material is in the form of carbohydrate, which is either mannitol or laminarin. True. Next, the major pigments are chlorophyll A, C, carotenoid, xanthophyll and fucoxanthin also, right. Next, vegetative cells have cellulosic cell wall usually covered outside with gelatinous coat, that is algin. Exactly true. So, the correct answer will be A, C, D and E, that is option 3. So, you have to remember that chart in the plant kingdom where it gives the complete features of Chlorophyceae, Phaeophyceae and Rhodophyceae. Keep that in your mind. Next, identify the step in tricarboxylic acid cycle which does not involve oxidation of substrate. Okay, which doesn't involve oxidation of substrate. If you look into the complete TCA cycle, you can clearly see succinyl CoA to succinic acid is mediated or catalyzed by the enzyme thiokinase, that is catalyzed by the enzyme thiokinase. Okay, and that will give out either one molecule of GTP or one molecule of ATP. Okay, so here the answer is 3. Next, statement based question from plant physiology. In C3 plants, some oxygen binds to rubisco, hence carbon dioxide fixation is decreased through by increasing the photorespiration fixation, CO2 fixation will be decreased. Next, in C4 plants, mesophyll cells show very little photorespiration while bundle sheath cells do not show photorespiration. Wrong. In C4 plants there is no photorespiration at all. Okay, in C4 plants there is no photorespiration at all. So, here statement 1 is true and 2 is false. Option 3 is the correct answer. Next, this is from biomolecules. GLUT4 is a insulin is a hormone, trypsin is an enzyme. Okay, collagen is intercellular ground tissue and GLUT4 enables glucose transport. So, A is 4, B is 1, C is 2, D 3 is option 1 is the correct answer. Next, DNA present in the chloroplast. DNA present in the chloroplast, if you see this in the chloroplast structure, if you see both chloroplast and mitochondria have double-stranded circular DNA, same as that of prokaryotes. That is because those two organelles have evolved from the prokaryotes, where we learned clearly in the endosymbiotic theory, okay, wherein both chloroplast and mitochondria have 70S ribosomes and they have double-stranded circular DNA. That is why we call those two organelles as semi-autonomous or self-replicating organelles. Next, monodalphous, diadalphous, polyadalphous, epiphyllous. Okay, so these are different types of the stamens. One is showing adhesion and the rest 3 cohesion. Diadalphous, though, of course you will see it in P. Next, epiphyllous condition is in lily. Okay, so option 1, wherein A is 4 monoadalphous China rose and polyadalphous. So A is 4, next D is 3, A is 4, D is 3 and B is 2. A is 4, B is 2, C is 1, D is 3, option 1. A polyadalphous is in citrus. So monoadalphous China rose, diadalphous is seen in Fabaceae, that the best example is P. Polyadalphous citrus epiphyllous condition you observe in Liliaceae family, example is lily. Epipetalous condition you observe in Solanaceae. Next, which of the following are fused in somatic hybridization? In somatic hybridization, two protoplasts of two different cells are fused. Okay, so that is option 2. So, two protoplasts are fused in the somatic hybridization. Next, again match the following type of question from respiration in plants. Citric acid cycle or TCA cycle is operated in mitochondrial matrix. Glycolysis takes place in cytoplasm both for aerobic and anaerobic respiration. ETS, electron transport system, which is the last step or ultimate step of aerobic respiration, that is seen in inner mitochondrial membrane. Next, proton gradient is observed in intermembrane space of mitochondria. So, if you clearly see, Nana, A is 2, B is 1, C is 4 and D is 3, option 2 is the correct answer. And next, which of the following statement is correct regarding the process of replication in E. coli? Okay, the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 3 to 5 wrong. Always a new strand, new DNA strand or new RNA strand that is synthesized from 5 prime to 3 prime only. Synthesis is always from 5 prime to 3 prime only. So, here the DNA dependent RNA polymerase will catalyze the polymerization in one direction, that is from 5 prime to 3 prime. Option 2 is correct. Next, which one of the following is the correct product of DNA dependent RNA polymerase to the given template? So, for us the template strand is given, we have to find out the RNA strand. Just now we have discussed na beta, new strand is always synthesized from 5 prime to 3 prime direction, identifying the 3 prime end of the parent. So, here TAC means we will get AUG, because adenine will code for uracil in mRNA. In mRNA, adenine will code for uracil. Next, A, that is U, C, next adenine, so again it is uracil. Here itself you will get from 5 prime to 3 prime, that is option 3. AUG, UAC, next CG and triple U, and last also you can check like yes. So, option 3 is the correct answer. So, if you want to write it more clearly, just you can write it over here. So, it is from 5 prime to 3 prime, right? Yeah. So, here it will be what, Nana? Adenine, and here it will be uracil, guanine. Adenine codes for uracil, A, C, C, G, U, 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 T. Again it is coding for A, U, A, G, G. A is coding again for U and T complementary, that is A, G. A is coding again for U till 3 prime. So, it is as it is of this, right. So, in this way you can decode. Look into what strand he is asking you. Here he has provided template and asking you for mRNA. Sometimes mRNA code is given and asked for coding strand. Sometimes coding strand is given and asked for mRNA. Always do remember, mRNA strand will be complementary to the template strand and same as that of coding strand, except in the place of T in the coding strand. Here in mRNA it will be uracil. That's it. Okay. Next. Yeah. Here if you clearly see in the cell cycle, Nana, diakinesis, all the 5 substages of prophase are given. This is utmost important. Don't miss that at all the 5 stages in the cell cycle and cell division, wherein diakinesis, okay, diakinesis, which is the last stage. In the last stage what happens? Completion of terminalization of chiasmata, pachytene recombination nodules, zygotine synaptonemal complex formation, liptotine chromosomes will look like thin threads in the liptotine. So, D is 3, C is 1, B is 4 and A is 2. Option 3 is the correct answer. Next question, if you look into, Nana, next question. Yeah. Like again, this is from biomolecules, wherein enzymes are given and their bonds are asked. Lipase, ester bond, okay, nuclease, phosphodiester bond, protease, peptide bond, amylase, glycosidic bond. So here what is the answer? A is 2, B is 4, C is 1 and D is 3. Option 3 is the correct answer. Next, this is from Cell the Unit of Life again. Axoneme is the core part. If you look into flagella or cilia, which originates from cell membrane and they are the extensions of cell wall. So, if the central portion of this flagella or cilia is what? Axoneme. And cartwheel pattern is seen in the centriole. Cristae is seen in mitochondria. Axoneme is seen in cilia and flagella. A is 2 and cristae is 4. Okay, just a second. C is 4 and B is 1, A is 2 and D is 3. Okay, satellite DNA is present in the chromosome, which is the secondary constriction of the chromosome. Okay, so here it is option 4. And next, following are the stages of cell division. Okay, we have 4 stages of cell division, right? First is G1 phase, next S phase, next G2 phase. In M phase we have karyokinesis and cytokinesis. Let me draw one cell cycle. So, if you clearly look into here, beta, first is G1 phase followed by G2 phase followed by S phase, then it is M phase. M phase has karyokinesis and cytokinesis. Okay, karyokinesis and cytokinesis. So, here the very first stage is G1 phase followed by S phase, next G2 phase, then karyokinesis and cytokinesis. So, E, C and then A followed by D and B. Option 4 is the correct answer. You have to remember the sequence of the stages in cell cycle for sure, all the phases of cell cycle. Next, this is from again principles of inheritance, genetic disorders. Down syndrome is a chromosomal disorder which is caused due to trisomy in 21st chromosome. Next, alpha thalassemia is because of 11th chromosome mutation, and beta thalassemia is because of the mutation in the short arm of 16th chromosome. Next, Klinefelter syndrome is because of the trisomy of XXY which is seen in males. Okay, trisomy of allosome, that is Klinefelter syndrome in males. So, if you look into the options carefully, beta, A is 3, B is 1, just a second, alpha thalassemia is of 11th chromosome, yeah, Down syndrome, it is of 21st chromosome, so it is of option 3. Clearly note down, A is, I think it is not given here, A is 3, B is 1, C is 4 and D is 2. Okay, this is the right answer. Next, mitochondria and chloroplasts are double membrane bound organelles. True. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Wrong. Okay, inner membrane of mitochondria is less permeable as compared to the chloroplast. Chloroplast will have more permeability. Once again read out the statement. Inner membrane of mitochondria is less permeable as compared to the chloroplast. True, okay, because chloroplast inner membrane is more permeable, mitochondrial membrane, inner mitochondrial membrane is less permeable because it has more transport pumps and channels. Okay, it will not allow that much permeability when it comes to inner mitochondrial membrane, whereas outer mitochondrial membrane is permeable. So here the correct statements, both statement 1 and statement 2 are true, okay, which is option 1. Next question, if you look into Nana, this is from ABO blood grouping, where he says that the blood group of father is B positive, blood group of mother is A positive, child is O positive. Their respective genotypes he is asking, okay. Respective genotypes will be, if child is O positive, definitely the father's blood group will be IB only, and mother's blood group will be IA and IO. Then only there is a possibility for the child to have O blood group, okay. So here what will be the genotype? Simply looking into the question we can say, right, IB I gene, IA I gene and IO, option A, okay. So 1 is the correct answer. Last question, if we look into, again this is from Mol Bio, molecular basis, wherein match the following type of question is asked. RNA polymerase 3, okay, RNA polymerase 3 is coding for, will synthesize, RNA polymerase 3 will synthesize snRNA and tRNA. Always in every NEET previous year question, there is a question on RNA polymerase 1 or 2 or 3. So don't miss revising RNA polymerases of a eukaryotic cell. Next, termination of transcription is done by Rho factor, okay. RNA polymerase along with Rho factor will initiate termination process in transcription. Splicing of exons is done by SNRPs, okay. Splicing of exons in eukaryotes is done by SNRPs. Next, Tata box is at promoter region. So here what is the right answer, Nana? A is 4, A is 4, B is 3, C is 1 and D is 2. So option 4 is the correct answer. So with this we have finished all the NEET botany previous year questions. In the coming classes we are having mega marathon sessions of all the subjects in our channel. So stay tuned and be focused on your preparation. Thank you.